Welcome back. This session is devoted to the transport of oxygen and carbon dioxide by the blood, and it's often considered after pulmonary gas exchange because in a sense it's the next link in the movement of oxygen from the atmosphere to the mitochondria. But we're going to look at it now because in talking about diffusion and pulmonary gas exchange, we need to use concepts such as the oxygen dissociation curve, and so it makes a lot of sense to talk about them now. And I'm going to start by looking at some of the important concepts having to do with the gases, the concentrations, partial pressures, and so on. And here we have a sample of air, dry air, at sea level, and the concentration of oxygen is 21%. That means that of all the molecules, 21% of the molecules are oxygen molecules. So the concentration of oxygen in dry air is 21%. And we can also show that using this nomenclature here with the square brackets, concentration of oxygen. We can also refer to it as 21 ml per deciliter. Same thing. Now, percentage concentrations all add up to 100. But in some cases, it's more useful for us to use what are called fractional concentrations. And the only difference is that the fractional concentrations add up to 1. So the fractional concentration of oxygen then would be 0.21, and you can see that all the fractional concentrations, all the gases, would add up to 1. Now let's look at the notion of partial pressure. It's a very important idea. We use it extensively. And the partial pressure is simply the oxygen concentration, in this case the fractional concentration, multiplied by the barometric pressure, which at sea level is 760 millimeters of mercury. So we multiply 0.21 times 760, and that gives us a partial pressure of oxygen in dry gas of 160 millimeters of mercury. Now there's a minor complication that we should deal with here, and that is the effects of water vapor. Because when air is inspired into the lung, it is warmed and moistened and saturated with water vapor at the prevailing temperature of 37 degrees Celsius. And at that temperature, the partial pressure of water vapor is 47 millimeters of mercury. Now that only depends on temperature. It doesn't depend, for example, on barometric pressure. You go to the summit of Mount Everest where the barometric pressure is much less, but the partial pressure of water vapor at 37 degrees Celsius is still 47. That's very important. As a matter of fact, if you increase the temperature to 100 degrees Celsius, then the water vapor pressure is 760. And that is the reason why water boils at that temperature of 100 degrees Celsius. So in order to calculate the partial pressure of oxygen in moist inspired gas, we have to subtract the water vapor partial pressure, which is 47. We subtract that from 760, that gives us the partial pressure in the dry gas of 713, and therefore the partial pressure of oxygen is 0.21 times 713, which comes out to 150 millimeters of mercury. So that's the partial pressure that we're actually dealing with. The organism, if you like, is exposed to a partial pressure of 150 millimeters of mercury in the moist inspired gas. Now let's look a little bit further at the effects of dissolving the gases in liquids. And on the left here we have a cartoon showing air, moist inspired air at 37 degrees C has a PO2, as we've seen, of 150 millimeters of mercury. And we allow that air to become equilibrated with water below it. By equilibration we mean we wait until the number of oxygen molecules entering the water is the same as the number escaping. See, we've got equilibrium. And the question is, what is the partial pressure of the oxygen in the water? The answer is that it's the partial pressure of the gas in equilibrium with the water. Now that might sound like a rather simple thing, a simple concept, but actually initially some students get confused about this. For example, they'll be faced with blood, arterial blood that has a PO2 of 100, and initially they might ask questions like, well, is that caused by the dissolved oxygen or is it caused by the oxygen linked to hemoglobin? Not useful questions. The only way we can think of the partial pressure of a gas in a liquid is the partial pressure of the gas in equilibrium with the liquid. So that's a very important concept that we've talked about here. Now we saw that the concentration of oxygen in air is 21 mils per deciliter. What about the concentration of the oxygen in the liquid? Well, it's very much less. And that, of course, is because the solubility of oxygen in water is relatively low. In fact, the solubility is shown down here as 0.003 mils per deciliter per millimeter of mercury. And so the amount of oxygen at a PO2 of 150 is going to be 150 times 0.003, and that comes to 0.45 mils per deciliter. Okay, so that's oxygen in physical solution. Now you may say, well, why can't we use oxygen in physical solution in the body to provide the required amount of oxygen to the periphery? And a simple calculation will tell us that that's impossible. Let's look at it this way. The solubility of oxygen, we said, was 0.003 mils per deciliter per millimeter of mercury. PO2 in arterial blood, which we haven't really discussed yet, we will extensively eventually, but take it from me that it's about 100 millimeters of mercury. And therefore the oxygen dissolved in arterial blood is going to be 100 times 0.003 or 0.3 mils per deciliter, which is equal to 3 mils per liter. Now suppose the cardiac output during heavy exercise is 30 liters per minute. That means that the maximum oxygen available is going to be 90 mils per minute. However, the tissues require about 3,000 mils per minute of oxygen, 3 liters per minute maximal oxygen uptake. So obviously the oxygen in solution is not sufficient. There's got to be another mechanism for oxygen transport, and of course, as we all know, this is hemoglobin. Just as a matter of light relief, there are animals that exist without oxygen-carrying pigments, and one of them is the Antarctic ice fish. The Antarctic ice fish has no hemoglobin. And you may say, well, how does it get on? Well, I suppose maybe it's a bit lazy, but the other thing is that it is at a very cold temperature where the solubility of oxygen is greatly increased. However, I wouldn't recommend this particular evolutionary solution. I think we've got a better one with our use of hemoglobin. Now, I'm not going to talk a lot about the structure of hemoglobin because it's really the province of the biochemists, and it's an extremely interesting subject, but let's just review it very briefly. Hemoglobin, the hemoglobin molecule, first of all, has a heme compound, which is an iron porphyrin compound, and this is linked with the globin, which has 4 polypeptide chains, 2 beta chains, and 2 alpha chains. And the whole thing has 4 heme molecules in it, as you can see, and those are the binding sites for oxygen. Now, there are various types of hemoglobin, and again, I'm not going into them in detail here. The most important is hemoglobin A, adult hemoglobin, which is the one that we're using right now. Another is hemoglobin F, fetal hemoglobin, which is used by newborns or also in utero, and is gradually replaced by adult hemoglobin after a few months of birth. The important thing about fetal hemoglobin is that it has a very high oxygen affinity, which is a very good thing because the fetus lives in a very low oxygen environment. There's also a hemoglobin known as sickle cell hemoglobin that we should mention. In that particular hemoglobin, one of the chains is altered slightly because of mutation, and it turns out that at low oxygen concentrations, the hemoglobin tends to crystallize in the cell. The cells develop all sorts of bizarre shapes, as you can see here, that they're called sickling as a result, and they also form small thrombi, and it's a very unpleasant disease as a result. Now, let's talk about the oxygen dissociation curve, which I'm sure most of you are familiar with to some extent. Here's the oxygen dissociation curve. And first of all, how would we develop an oxygen dissociation curve? Well, what we do is we would take some glass vessels, known as tonometers, put a small amount of blood in the bottom, and then above the blood we would put gases of different PO2 values. And we would then allow equilibration to take place for each of these gas experiments. We would then remove the blood, measure the oxygen concentration, subtract the amount that was dissolved, because we know what the solubility of oxygen is, and the rest of it would be oxygen combined with hemoglobin. And what we're seeing here is a nice plot of hemoglobin saturation against PO2 with oxygen concentration on this axis. Now, the hemoglobin saturation is simply the percentage of the oxygen binding sites that are bound with oxygen. The oxygen concentration, of course, is the mils of oxygen per deciliter, per 100 mils of blood. You can see on this that the amount of dissolved oxygen is very small, up to PO2s of arterial blood, about 100. If, however, as shown here, we increase the PO2 to about 600, which can occur in arterial blood when the subject is breathing 100% oxygen, then you can see that the amount of dissolved is substantially increased. But generally speaking, the dissolved oxygen is a very small fraction of the total oxygen in the blood. Now, the total amount of oxygen that the blood can carry attached to hemoglobin is known as the oxygen capacity. Okay? And it's also an important term. Now, the hemoglobin oxygen dissociation curve, as we can see, has an S shape with a small curve at the bottom here and then a long curve at the top. And the shape has to do with the change in configuration of the molecule as the oxygen is loaded. It's rather difficult to get the first molecule of oxygen onto the hemoglobin, but once the first one is on, it's easier to oxygenate, and that gives us the curve here. The shape of the oxygen dissociation curve has a number of advantages. One of them is that if we go to high altitude and the PO2 falls substantially, say to 60 millimeters of mercury, we still have a very large amount of oxygen attached to the hemoglobin. So this flat or nearly flat upper region is very valuable from that point of view. This part, the part with the steep part of the curve, is also useful because in venous blood, when you extract oxygen, the fall in PO2 for a given amount of oxygen extracted is relatively small. So that's also advantageous. Now let's go to the next slide, which shows the anchor points of the oxygen dissociation curve. And there are three that I like to remember. They're not difficult to remember. One is that in arterial blood with a PO2 of 100, we have an oxygen concentration very nearly 100%. It's about 97%. So in arterial blood, the hemoglobin is almost fully saturated. Then we go down to what we call the mixed venous blood. That's the blood in the pulmonary artery coming into the lung. That has a PO2 of about 40 and an oxygen saturation of about 75%. And then we also, another useful anchor point is what's called the P50. The P50 is the partial pressure of oxygen for 50% saturation. And in normal arterial blood, the value is about 27. So these three anchor points are quite useful if someone says to us, well, this person's got a PO2 of 50, for example, in the arterial blood. Well, you know that it's going to be somewhere up in this range here, around about 80%, the saturation around about 80%, because we know that with a PO2 of 40, saturation is 75%, so that gives us a sort of ballpark figure if someone indicates what the PO2 is and we wonder what the oxygen saturation is. Now it's very important to understand how we calculate the total oxygen in blood, because we need to do that from time to time. And here we have an equation, not a difficult equation, showing us how to calculate the total oxygen concentration of blood. That's going to be the oxygen attached to hemoglobin, which is the line here, and the oxygens dissolved. Now, how much oxygen can be combined with hemoglobin? Well, it turns out that 1 gram of hemoglobin can combine with 1.39 mils of oxygen. Incidentally, sometimes you'll see this number given as 1.36, particularly in the older literature. And the reason is that when people tried to measure it, there was always a little bit of hemoglobin that was spoiled. It was methemoglobin or something like this, and it wasn't combined with oxygen. But when we know the molecular weight of hemoglobin, as we do now, we know that the true value is 1.39 mils of oxygen per gram of hemoglobin. And then we have the hemoglobin concentration of blood, which is, ballpark figure, about 15 grams per deciliter, 15 grams per 100 mils. So in other words, the total oxygen concentration bound to hemoglobin is going to be 1.39 times, say, 15 times the oxygen saturation, which in arterial blood is, as I said, about 97%. So this would be 97% over 100, 0.97, plus the amount that's dissolved. So let's just do this calculation for arterial blood, for example, where the PO2 is, say, 100 millimeters of mercury. This would be 1.39 times 15 times 0.97 plus solubility 0.003 times 100, the PO2, and that all comes out to be about 20.5 mils per deciliter. Now it's important to understand the relationships, to be clear about the relationships between PO2, oxygen saturation, and oxygen concentration. Because in physiology generally, and certainly in clinical medicine, it's important to understand how various abnormal conditions affect these variables. So let's look at some of them here. Let's take normal blood with a hemoglobin concentration of 15. And you can see that at a PO2 of 100, which is around about here, it has a saturation of nearly 100%, and it has an oxygen concentration of something like 20 mils per deciliter. Now, if we increase the hemoglobin concentration to 20, that's called polycythemia, happens, for example, at high altitude. Now you can see that for a normal PO2 of 100, the saturation is still 100, but the oxygen concentration is very much greater because, of course, of the increased amount of hemoglobin. By the same token, if we have anemia where the hemoglobin concentration is reduced to 10 grams per deciliter, then at a PO2 of 100, the saturation again is very nearly 100, 97%, but of course the oxygen concentration is less. Take another example. Suppose we go to high altitude and we reduce the arterial PO2 to, say, 60. Then what we'll find is that the oxygen saturation will be reduced and the oxygen concentration will be reduced together with this reduction in arterial PO2. Now, the oxygen dissociation curve is shifted under various conditions, and these are very important. Now, we talk of shifts of the oxygen dissociation curve. We could also talk of changes in the oxygen affinity of the hemoglobin. Because if we shift the curve to the left, for example, the concentration of oxygen, the saturation or the concentration for a given PO2 is increased. So that means that we've increased the oxygen affinity of the hemoglobin. And it turns out that there are 4 factors that shift the curve to the right or reduce the oxygen affinity of the hemoglobin. Those are temperature, PCO2, hydrogen ion concentration, and 2,3-diphosphoglycerate. So if we have an increase in temperature, an increase in PCO2, increase in hydrogen ion concentration, the curve shifts to the right, and the various plots are shown here. Now you may find it difficult to remember that, but I don't, because I remember just one thing, and that is in exercising muscle, the temperature increases because of the metabolism, the PCO2 increases because the exercising muscle is producing lots of CO2, the hydrogen ion concentration increases because of the increase in PCO2 and perhaps an increase in lactate as well, and it's advantageous in exercising muscle to have a right-shifted curve where the oxygen affinity is reduced, because that means that you can unload more oxygen for a given PO2 if the curve is right-shifted. 2,3-DPG is a product of red cell metabolism, and if that is increased, the curve is shifted to the right. What happens sometimes in stored blood is that the concentration of DPG is depleted, and that means the blood has a high oxygen affinity, and that makes it difficult to unload in the periphery of the body. Okay, so those are shifts in the oxygen dissociation curve. I'm now going to move from oxygen briefly to the transport of carbon monoxide. We're not going to say a great deal about this, but it is important because, as I'm sure you know, carbon monoxide is a very toxic gas, and it's important for us to understand what's happening when CO is inhaled. And this slide will probably confuse you initially, because we're showing the oxygen dissociation curve here, which we're familiar with, but we're also showing the dissociation curve for carbon monoxide. You may say, well, it doesn't look like an it doesn't look like a dissociation curve at all. It looks like a box with a corner. Well, the fact is, dissociation curve for carbon monoxide is actually the same as oxygen. But what we've done is we've greatly compressed the x-axis so that for a very small partial pressure of CO in blood, the saturation is almost 100%. In other words, carbon monoxide has an extremely high affinity for oxygen, about 240 times the affinity of oxygen. So if you change this scale here, if you magnified it by 240, you'd get the dissociation curve of carbon monoxide. Of course, that's very important because it means that if you have a small partial pressure of carbon monoxide in the blood, it is able to tie up, to combine with a large amount of the oxygen. And that means that there's only a small amount of oxygen left over for normal metabolism. So that's a very important feature. Another important feature, which is actually one we saw on the previous slide, although I'm going to leave it here to talk about it, is the fact that if you have 1/3 of the hemoglobin bound to carbon monoxide, as is the case here, so that the available hemoglobin concentration is only 10 grams per deciliter, what's available for oxygen carriage, then it turns out that the curve is shifted to the left. In other words, the affinity of the hemoglobin for oxygen is greatly increased. And this makes it difficult to unload the oxygen in the periphery, and that's another reason why carbon monoxide is a very toxic gas. Now let's move to the transport of carbon dioxide in the blood, which is the other major gas, of course, of respiratory interest. And carbon dioxide is carried in the blood in three forms. One as dissolved, secondly as bicarbonate, and third as carbamino compounds. So let's look at these in turn. It turns out that the solubility of carbon dioxide in blood is substantially greater than that of oxygen. We saw that the solubility of oxygen was only 0.003 mils per deciliter. The solubility of carbon dioxide is about 0.067. That's about 24 times greater than the solubility in oxygen. And it turns out that as a result, the carriage of carbon dioxide in the blood, the dissolved carbon dioxide plays an important role. You can see in arterial blood here that about 5% of the carbon dioxide being carried is in the dissolved form, and that if you look at the difference between the mixed venous blood and the arterial blood, in other words, when the CO2 has been evolved into the alveolar gas, about 10% of the amount of CO2 is dissolved CO2, which has been evolved. So dissolved carbon dioxide is quite important, much more important than dissolved oxygen. Let's look at the very important subject of the formation of bicarbonate. When carbon dioxide is added to water, it forms carbonic acid. Now, under normal conditions, this is a very slow reaction. But in the red cell, there is an enzyme called carbonic anhydrase, which enormously accelerates this reaction. And this turns out to be critical in the carriage of CO2 in the blood, as we'll see shortly. The carbonic acid immediately dissociates into hydrogen ions and bicarbonate ions, and of course this does not require an enzyme. So these are the steps required in the carriage of carbon dioxide in the form of bicarbonate. Let's turn to carbamino compounds. It turns out that carbon dioxide can link with the terminal amine group of proteins in the blood. And the most important protein by far is hemoglobin. So carbon dioxide links with hemoglobin in this way and forms what is called carbaminohemoglobin. And there are carbamino compounds with other proteins in blood as well, but they're not nearly so important. Now the next slide looks pretty daunting at first, doesn't it? But it's actually a very useful way of pulling together all the factors involved in CO2 transport in the blood. And I'm going to go through it slowly, and I think you'll see that it makes a lot of sense, even though initially it looks very complicated and daunting indeed. And what we're looking at here is the uptake of CO2 by the blood in the periphery of the body. For example, in an exercising muscle. The muscle is producing CO2 because of metabolism, and some of that CO2 then will diffuse across the capillary wall in the leg, for example, or whatever. Some of it then will be dissolved in the plasma, a relatively small amount, but some will be. But none of it in the plasma will form bicarbonate, because you need the enzyme carbonic anhydrase to do that, and the plasma does not contain any. So not very much occurs in the plasma itself. But once the CO2 diffuses into the red blood cell, shown by this great big circle here, then we can form bicarbonate and the other things as well. First of all, we should say there is, of course, some carbon dioxide that's dissolved inside the red cell, but then a substantial amount of it combines with water under the effect of the enzyme carbonic anhydrase to form carbonic acid. And this dissociates then into hydrogen ion and a bicarbonate ion. Now it turns out that these hydrogen ions are mopped up by the reduced hemoglobin in the red cell. In other words, the reduced hemoglobin is able to combine with these protons, these hydrogen ions, much more easily than the oxygenated hemoglobin. So you can see that having reduced hemoglobin in the red cell then accelerates the formation of carbonic acid. And this means that the lower the oxygen saturation of the blood, the better able it is to take up carbon dioxide. Now, the bicarbonate ion builds up inside the red cell, and therefore some of it diffuses out into the plasma. But the hydrogen ions cannot diffuse out because the membrane is relatively impermeable to them. And so in order to maintain electrical equilibrium, some chloride ions have to move back. These are also negatively charged ions, have to move back into the red cell. And that's sometimes called the chloride shift, and that's necessary to maintain the equilibrium, that's part of the Gibbs-Donnan equilibrium in the red cell here. Now the oxygen, of course, is moving out, as we can see. Obviously the oxygen is moving out into the metabolizing tissue, and it turns out that these processes here increase the osmolar content of the red cell, and water tends to move in to the venous blood, but once the red cells go through the lung, the water moves out again. So let's turn to the third way in which the carbon dioxide is transported by the red cell, and that is, as we saw, by combination with hemoglobin to give carbaminohemoglobin. That is done, as I mentioned before, by a combination between the carbon dioxide and the terminal amine group of the hemoglobin. Now it turns out that the reduced form of hemoglobin, shown here, is much more able to form this carbamino compound than the oxygenated hemoglobin. So again, we have a different effect here, a different mechanism by which the reduced oxygen in the hemoglobin, the reduced deoxygenated hemoglobin, is better able to take up the carbon dioxide. So there are two effects here that make it easier for the red cell that has a reduced amount of oxygen in it to take up carbon dioxide, and this is known as the Haldane effect. Okay, so that's a summary of what happens within the red cell when the CO2 is taken up. It's a complicated picture perhaps, but if you go through it slowly as I've done, I think you'll see it makes sense, and it really puts together all the factors involved in CO2 transport by the blood. So let's just summarize the various forms of CO2 in the blood. You can see that in arterial blood, the vast majority of the bicarbonate ions of the CO2 transport is taken up as bicarbonate, carried by a carbonate. Only 5% is in the dissolved form, and only 5% is in the carbamino form. By contrast, if you look at the difference, the exchange of carbon dioxide in the lung or in the tissues, a substantial amount is taken up by carbamino and also dissolved, although still the largest amount is in the bicarbonate. Now let's look at what we call the CO2 dissociation curve. Now I should say that that's a rather loose way of talking. We use that term, of course, by analogy with the oxygen dissociation curve. Here we're looking at the relationship between carbon dioxide concentration in the blood and the CO2 partial pressure. So, you know, that's analogous to the oxygen dissociation curve. But purists will probably say, well, you shouldn't really call it a dissociation curve, but we will, because it's the simplest way of describing it. And you can see immediately that the CO2 dissociation curve is steep, and it's almost linear in the working range, quite unlike the oxygen dissociation curve, which has this marked S shape and is very nonlinear. Another thing you'll see is that the amount of CO2 that can be combined, the CO2 concentration, at a given partial pressure here, is greater when you reduce the oxygen saturation of the hemoglobin. We mentioned that before when we were talking about the uptake of CO2 by the red cell, and that is the Haldane effect. So that when you've got no oxygen in the red cell, you can take up an enormous amount of hemoglobin, but even mixed venous blood with an oxygen saturation of 75% can take up more than arterial blood with an oxygen saturation of 97 or so percent. And over here we've got what you might call the physiological route from the mixed venous blood to the arterial blood. Now we haven't come across the PCO2 in mixed venous blood yet, but it's about 47 millimeters of mercury. PCO2 in arterial blood is about 40. So this is the path that's taken by the CO2 when it's evolved in the lung, and you can see the fact that the curves are different for low oxygen and high oxygen blood makes this path even steeper. Let's have a last look at the oxygen and CO2 dissociation curves. We've got them both on the same axes here, and immediately you can see that the CO2 dissociation curve is much more linear and it's much steeper than the oxygen dissociation curve. And one of the results of that is that the difference in partial pressure between the arterial and the mixed venous blood is very much less for CO2 than it is for oxygen. In oxygen, you've got an arterial PO2 of about 100, mixed venous PO2 of about 40. By contrast, for CO2, the arterial PCO2 is about 40, and the mixed venous PCO2 is about 47. So there's a very small arterial venous difference for PCO2. In spite of that, of course, the amounts of oxygen and CO2 that are being exchanged in the lung or in the periphery are about the same. And we'll be talking about that much more in a short period of time. But although the arterial venous difference for concentration of oxygen or CO2, almost the same, the PCO2 difference are very striking. So let's just summarize some of the main points we've made. First of all, we pointed out that the importance of understanding the definitions of concentrations of gases and their partial pressures. Partial pressure of a gas is the fractional concentration times the barometric pressure. We then looked at the important effect of water vapor on partial pressure and showed that in moist inspired gas, the partial pressure of water, the water vapor partial pressure is about 47, and so that's got to be subtracted from the total barometric pressure in order to get the partial pressure of a gas in the moist inspired gas. We then looked at the very important topic of the partial pressures of a gas in a liquid, and we pointed out that the only sensible way of thinking about them is that the partial pressure of a gas in a liquid is the partial pressure of a gas in equilibrium with the liquid. We then looked at the carriage of oxygen by the blood. We pointed out that the oxygen in solution is nowhere near sufficient to look after the metabolic requirements, particularly during exercise, and therefore we have hemoglobin. We mentioned that the heme is an iron porphyry compound and that we have 4 polypeptide chains in the globin protein, 2 of them alpha and 2 of them beta. We talked about the way of deriving the oxygen dissociation curve, the advantages of its S shape, in particular that if you go to high altitude and your alveolar PO2 falls a little bit, then you lose very little oxygen in the blood. Another advantage in the periphery is that the dissociation curve is steep so that you can withdraw a lot of oxygen from the blood with a relatively small change in partial pressure. We referred briefly to the hemoglobins, the fetal hemoglobin and sickle cell hemoglobin, the latter being, of course, an abnormal one responsible for sickle cell disease. We briefly mentioned the importance of understanding the differences between the PO2, the oxygen saturation, and the oxygen concentration, because these vary in ways that initially may be confusing, but if you think it through, they're not all that difficult. We mentioned the factors affecting oxygen affinity, and we pointed out that reducing the CO2 increases the oxygen affinity, and I forgot to mention that this is known as the Bohr effect. Christian Bohr was a famous physiologist, and he was the person who has his name attached to this very important effect of CO2 in blood affecting the oxygen affinity. We then talked very briefly about the carriage of carbon monoxide in the blood, of course not a respiratory gas, a toxic gas, and pointed out that it has a very, very high affinity, 240 times the affinity of oxygen, and so it's a dangerous gas because it combines with a lot of the hemoglobin and doesn't leave it available for oxygen carriage, and it also incidentally shifts the oxygen dissociation curve to the left and makes it more difficult to unload oxygen in the periphery. We then turned to carbon dioxide transport and pointed out that this occurs in three forms: dissolved, as bicarbonate, and as carbamino compounds. We went into some detail in the way carbon dioxide is taken up by the blood in the periphery and pointed out that the lower the oxygen saturation, the more CO2 can be taken up, known as the Haldane effect. We also looked at the CO2 dissociation curve, showed that it's much steeper than that for oxygen, and it's also much more linear, and finally pointed out that this is the reason why, although the difference in CO2 and oxygen concentrations across the lung are very similar, the PCO2 difference is very much less than the PO2 difference. So that's the end of this brief discussion of the transport of oxygen and carbon dioxide by the blood, and we're going to go on next time to look at the acid-base effects of the CO2 in the blood. I'll see you then.